Psychiatric Society. I welcome you to yet another session of Brain on Wednesdays. The topic of the evening is glutamate dysregulation in psychiatric illnesses. To chair the session, we have Dr. Aparna Ramakrishnan, who has done her postgraduate studies from Father Muller Medical College and currently working at Kokilaben Dhirubhai Ambani Hospital, having had her stint with State GS Medical College, DY Patil Hospital and Medical College, as well as Grant Medical College and JJ Group of Hospitals. Since 2019, she has been associated with Dhirubhai Ambani Hospital at Bombay. Dr. Aparna, the floor is yours.
Thank you so much, sir. A very good evening to one and all. Thank you for the kind introductory words, sir. It is indeed an honor and privilege to be a chairperson for the second time on the Brain on Wednesday. And I would like to thank Dr. Shobha, who I have worked with, who's been my teacher when I worked in BRC Hospital, Dr. Rajkumar, of course, and Dr. Bhaskar Mukherjee, as well as Dr. Malaida. Now, the topic for today's discussion is...
Just a moment, Aparna.
Yes.
Samson, what number is it? SM351315F. Please mute yourself. Thank you. Go ahead, Aparna.
The topic for today's discussion is a very interesting one. Glutamate dysregulation in psychiatric illness. Now, all of us know that glutamate is the main excitatory neurotransmitter in the CNS. But at the same time, it also has a lot of peripheral involvement, especially in the autocrine, paracrine manner in pain signaling. And it is characteristically seen, glutamate dysregulation is characteristically seen in a neurodegenerative, neurodevelopmental, as well as psychiatric conditions like schizophrenia or Fragile X syndrome, or even it is even showing emerging roles in depression as well as anxiety. So without further ado, I would invite today's speaker, Dr. Sagar Bhalke, to shed some light on this topic. A few words about Dr. Sagar. He is an MD and DNB in psychiatry, and he's also a fellow in child and adolescent mental health at Nair Hospital, Mumbai. He's extremely passionate about medical biology and genetics, and this is his second time in Brain on Wednesday also. So I can't think of a better person to shed more light on this particular topic. So the floor is yours, Dr. Sagar.
Thank you, ma'am. Rajkumar sir, shall I share my screen now?
Yeah, go ahead.
Is it visible now?
Yes, but make it, yes, that's right.
That black box top right corner, just open it and just squeeze to the smaller one.
Yes, in the corner, right? Yes.
Finger. Because when you drag it, your cameras goes off.
Is this okay, sir?
Yes, it is okay.
Okay. So welcome to this session on glutamate. Basically, we'll be discussing about... Okay. So today, basically, we'll be discussing about the role of mental health disorders. This is an overview for today's session. Initially, we will see some chemistry and the evolutionary glutamate. After that, we'll be comparing with animal models, especially the rodents. And then we'll see some pathology. In pathology, we will be discussing about depression, schizophrenia, OCD, a little bit of autism, migraine, epilepsy. The major focus of this session will not be dementia, because we already know a lot of, we already know the role of glutamate in dementia, but it is the other mental health disorders we'll be focusing more on. Additionally, also cancer, because glutamate has a huge role in it also. And then the possible interventions, mostly pharmacological. So let us start with understanding the chemistry of glutamate. Synthesis of glutamate is de novo from glucose through TCA cycle. So we can see in this diagram here, glutamate is in equilibrium with alpha-ketoglutarate always, and is actively sequestrated into the storage vesicles presynaptically. And whenever a stimulation is required, the presynaptic vesicles secrete the glutamate, which enters into the synaptic vesicle and does its function. This is an important diagram which we need to understand before we go further with the glutamate details. So as you can see, presynaptically, glutamate enters into the vesicles and it is stored there. So glutamate is stored here. And then when stimulation is required, basically it is released into the synaptic vesicle. And from there, it enters into the postsynaptic neuron through three receptors, kainate, NMDA, and AMPA. All these three have same function to transport glutamate from here to postsynaptic neuron. And apart from that, one other structure which plays a major role is the astrocyte in glutamate regulation. So what astrocyte does is it has receptors called EAT, excitatory amino acid transporters. So what they do is they shift this glutamate inside the astrocytes. So clearance of glutamate is the role which is played by astrocytes. So any hyperactivity or any excess release of glutamate, it is taken care by the astrocytes. Along with that, astrocytes also produce GSH, glutathione, which is a antioxidant. So any inflammation happening in the synaptic cleft is also taken care by the astrocyte. So two roles by astrocyte. First is clearance of glutamate, and second is releasing of the antioxidants. I think this has already been covered by Nachiket Bhai in detail. So what are the major pathways in which glutamate is involved? All the primary systems use glutamate throughout the body, not just the brain. Retinal, ganglionic, I think there is some typing error here. Trigeminal nerve, spinal efferents, thalamocortical projections, pyramidal neurons of cortical limbic region, temporal lobe circuits, and the corticospinal tracts. Now let us understand the evolutionary basis or the evolutionary journey of glutamate. Glutamate is the amino acid and a neurotransmitter which is present in the maximum quantity. Around 60% of all the neurotransmitters is composed of glutamate. So initially it had a primordial role, basically a hub molecule in the transamination reaction. So involved in nitrogen metabolism, and gradually it also entered into the TCA cycle. Along with that, glutamate transporter started to evolve. There was only one glutamate receptor and transporter, and gradually they evolved into 1, 2, 3, 4, 5. So now we have multiple glutamate transporters and multiple glutamate receptors, increasing the complexity and the diversification of the function. We need to understand how glutamate works in rodents or other animals compared to humans, so that we get the context of complexity that the human glutamate system has. Human glutamate system has larger glutamatergic synapses. Humans have enlarged presynaptic active zones, more number of vesicles and larger presynaptic vesicles, broader postsynaptic density of the receptors, and this leads to basically higher quantal release of glutamate in the synapse compared to lower animals. There is stronger excitatory postsynaptic current and enhanced resilience to repeated activation. The increased size of vesicle basically ensures that there is higher glutamate released per stimulus coming from the afferent nerves. So what does this mean? This basically means that humans basically have a higher and more sustained state of transmission per stimulus, which is important because we perform higher cognitive tasks. We also know that astrocytes basically play a crucial role in glutamate metabolism. So astrocytes co-express glutamate dehydrogenase 1 and GDH2, which enable better glutamate oxidation into alpha-ketoglutarate. And whenever alpha-ketoglutarate is converted into glutamate, again it's released into the synapses. Enhanced calcium dynamics in human astrocytes leads to rapid modulation of the glutamatergic synapses. Now, all the diseases that we'll be talking about in the further 20 to 30 minutes will be mostly about the excitotoxicity, that is, increased amount of glutamate in the synaptic cleft. Very less focus will be given to low glutamate in the cleft, which does not lead to as severe impairment as high glutamate into the synaptic cleft. So excitotoxicity basically translates into higher glutamate into the synaptic cleft. NMDA receptor overactivation leads to neurodegeneration in the hippocampus and the prefrontal cortex, which are the regions central to memory and mood regulation. High glutamate in the cleft is associated with panic-like responses and behavioral rigidity in animal models. Excitotoxic stress also causes mitochondrial fragmentation and ATP depletion because the TCA cycle has to work more and more, leading to motivational anergia and psychomotor slowing. Very commonly seen in depression, anhedonia, sickness behavior. And chronic excitotoxicity alters the dendritic spine morphology, reducing the behavioral adaptability and emotional resilience. Going ahead with more about the metabolism.
If I may just interrupt, Sagar, you are not seen on the screen.
Just a minute. I think I have forgotten to remember the video is on. Am I visible now?
Person is
Yes.
not in a relationship.
Yes, you're visible.
Okay. Okay, so now let us discuss the redox state and the neurobehavioral outcomes of it. Glutamate works via system XC. Basically, system XC is an antiporter. We can see it in this diagram. So basically, this is the system XC, which we can see here. Basically, it functions in the reverse way of the EAAT transporter. Instead of taking the glutamate inside, it actually releases the glutamate outside and takes up the cysteine. And cysteine is used for creating antioxidants in the cell. Okay. So glutamine via system XC, that is an antiporter, controls the cysteine uptake for glutathione synthesis, a key antioxidant buffer. Redox failure enhances the NMDA sensitivity and stress vulnerability. To talk more on in more detail about this, basically what does redox failure means? Redox failure means that the antiporter is not working well, and therefore cysteine uptake is not there. And if cysteine uptake is not there, antioxidants are not produced. And if antioxidants are not produced, then NMDA sensitivity increases, leading to neuroinflammation. So this is associated with behavioral phenotypes like emotional volatility and irritability, which correlate with the glutathione depletion in VMPFC and amygdala. So what we can do is basically it is desirable that in the synaptic cleft, more antioxidants should be present. And if antiporter is not able to do that, we have to artificially incorporate antioxidants in the cleft. So one of the most common antioxidants that we know and we use is N-acetylcysteine. Basically, N-acetylcysteine will give away cysteine, and cysteine is used for production of antioxidants. Apart from N-acetylcysteine, vitamin C is also used for the same function. Lipoic acid is also used for the same function. Selenium is also said to play a role as an antioxidant. And hence it has shown promise in treatment of OCD, addiction, and schizophrenia. Now going ahead, we'll be discussing 3 to 4 major psychiatric diseases in relation to glutamate: schizophrenia, depression, OCD, and autism. So schizophrenia. Basically, there is NMDA hypofunction, which leads to disruption of the gamma oscillations, reducing working memory and sense of reality. So gamma oscillations basically are the oscillations which are produced in a frequency of 30 to 100 hertz in the brain. And these oscillations are a result of stimulation of GABA neurons. So once GABA neurons are stimulated, it leads to formation of gamma oscillations. And the stimulation of GABA neurons is caused by NMDA. So if NMDA is not functioning well, there is no GABA stimulation and there is no gamma oscillation. What happens if there is no gamma oscillations? There is reduced working memory, there is impaired sense of reality, which means basically delusions and hallucinations. There is also impaired glutamate on the paravalbumin interneurons, leading to poor sensory prediction and hallucination. So we can see the impact of gamma disruption. Basically, this is not impact of gamma oscillation disruption. So reduced working memory, reduced sensory binding, leading to visual hallucination, reduced cortical network coherence, leading to negative symptoms and disorganized thinking we see in schizophrenia, and reduced signal to noise ratio. So more noise and less signaling, leading to derealization or psychotic symptoms. So this was the role of glutamate and NMDA in schizophrenia. Along with that, there is also a reduced EAAT in astrocytes. And we already saw that EAAT is useful for clearance of excess glutamate in the cleft. So if there is reduced EAAT in astrocytes, it may exacerbate glutamate imbalance across dorsolateral prefrontal cortex, linked to negative symptoms. Antipsychotic non-responders often show persistent glutamatergic hyperactivity in anterior cingulate cortex on spectroscopic scans. This results in disinhibition of excitatory pathways, disrupting thalamocortical and corticolimbic circuits. Hence, NMDA receptor antagonists such as memantine and ketamine, basically not memantine is too weak, but ketamine. So whenever we use ketamine in normal people, it mimics some symptoms of schizophrenia in healthy individuals, further supporting the above model. Let us talk about depression. In depression, basically there is astrocytic loss. Nachiket Bhai covered this in the last session, and also glutamatergic collapse. So, glutamate levels are often reduced in the anterior cingulate cortex, correlating with the psychomotor retardation and affective blunting. But it is not always that the glutamate levels are reduced in depression. There are some cases where the levels are higher also. And whenever high, it leads to symptoms of irritability, anger spells, and in severe cases also suicidal activity and tendencies. Astrocytic atrophy in hippocampus and PFC comprises glutamate-glutamine cycling and neurotropic support. If EAT downregulation is there, it leads to inefficient synaptic glutamate clearance, as we saw already. And if there is no good clearance, it results in cortical fuzz, a noise signal linked with cognitive distortions, rumination, and a cognitive dulling, which we see in depression. EAT activators basically will ensure that there is good glutamate clearance, such as the riluzole. So riluzole shows promising treatment-resistant depression by enhancing glutamatergic tone, glutamatergic clearance, and synaptic integrity. Normally, riluzole is used in ALS, but now studies have shown that it can be used, it can be repurposed in treatment of depression. Antidepressant effects of ketamine are thought to stem from NMDA receptor blockade, causing a rapid glutamate burst that activates AMPA receptors and enhances the synaptic connectivity via BDNF release. So basically, ketamine acts on NMDA, blocking it, leading to BDNF release and synaptic plasticity. So the previous two slides showed that in depression, if we are able to clear the glutamate, which is excess in the synaptic cleft, it can lead to betterment of symptoms. But then some researchers thought to check it objectively. And what they did is in around 60 to 70 patients, they checked before antidepressant and after antidepressant level of glutamate with glutamine in the synaptic cleft. And surprisingly, they found out that after treating with antidepressants, there was no statistically significant association between pre-treatment glutamate GABA and post-treatment glutamate and GABA. So conclusively, they told that glutamate, GABA, and the ratio is not predictive biomarker for SSRI or any kind of treatment to MDD. So although glutamate has some role in treatment of depression, it is not basically not whole and sole the reason for symptoms of depression. Glutamate and OCD. Okay. So now let us see OCD. So what is behavior in OCD that we commonly see? Excessive repetitive grooming behavior in animal models. It basically mirrors the compulsive symptoms seen in the human OCD. And it is strongly related with the synaptic scaffolding protein, SAPAP, basically synapse associated protein 90. So OC symptoms are related to this scaffolding protein. And we also know that OC symptoms are treated with antidepressant. And OC symptoms are related with high glutamine, is what the circuit level mechanisms have shown, hyperglutamatergic state. So we saw that SAPAP is not working, antidepressants are treating it, and also OC symptoms are due to high glutamine. This led to a conclusion by the researchers that maybe SSRIs in some way are altering glutamate. And therefore, there is a glutamate serotonin crosstalk in the mediating, which is mediating the compulsive behavior. And hence the role of antidepressants in reducing glutamate and treating OCD. Bipolar disease, this slide is just namesake because schizophrenia, bipolar, both are same. But we have seen elevated glutamate and glycine, glutamine levels reported during manic phases. And lithium and valproate modulate glutamate receptor function and decrease the excitatory transmission. Going ahead, seeing neurodegeneration in other neurological disorders. Excess glutamate leads to excitotoxicity, which is because of overactivation of NMDA receptors, allowing for excess calcium influx, and which finally leads to mitochondrial damage, oxidative stress, and apoptosis because of the free radicals released by mitochondrial damage. This is central in few diseases such as Alzheimer's disease, which is linked to A beta induced dysregulation of glutamate transporters and NMDA receptors. Parkinson's disease, where there is altered glutamatergic drive onto basal ganglia circuits, and Huntington's disease, where striatal medium spiny neurons are vulnerable to glutamate mediated toxicity. Some points related to gut-brain axis also. So basically, gut-brain microbes, they regulate glutamate metabolism via glutaminase and glutamine synthetase pathways. Glutamate affects the vagal tone and HP axis sensitivity, which are key in stress disorders. Dysbiotic microbiomes may increase the glutamate levels, which is undesirable, and which crosses the leaky blood-brain barrier and exacerbates the neuroinflammation. So, fecal transplant studies have shown that behavioral changes are linked to altered glutamate receptor gene expression in the hippocampus. Role of glutamate in autism spectrum disorder. So, there is an imbalance in the glutamate-glutamine ratio. The spectroscopic studies have shown elevated glutamate in the synaptic cleft and altered ratio in the anterior cingulate cortex and basal ganglia of individuals with autism spectrum disorder. This suggests that there is hyperglutamatergic tone in the cleft, impairing the synaptic homeostasis and plasticity. Along with that, there is AMPA receptor deficit also. So clearance of glutamate through cleft is not there, implicating excitatory synaptic deficits that disturb the motor coordination and learning. Also, the transporter downregulation is also there, and ratio imbalance is also there. Excitatory and inhibitory imbalance is also there. A decreased GABAergic signaling in somatosensory cortex leads to hyper-responsivity to sensory stimuli and may underlie the tactile defensiveness and somatic symptoms that we usually see in autism spectrum disorder. These are some other points related to autism. Maybe I'll share the PPT later. Okay. There are a few principles related to neurotransmitter that we should know. Most importantly is the Dale's principle, which suggests that one neuron only secretes one type of neurotransmitter. But now this fact or principle has become obsolete. And we now know that many neurons co-release more than one neurotransmitter at a time. Examples being dopaminergic neurons of ventral tegmental VTA co-releasing glutamate, Cholinergic neurons also releasing GABA and acetylcholine. Thus, modern neuroscience says that Dale's principle was old, and now one neuron can secrete more than two different types of neurotransmitters. So, based upon that, now understand the dynamics between glutamate and GABA. Basically, researchers have found out that GABA and glutamate are stored in the same neuron, in the same vesicles, but they are released in a biphasic miniature postsynaptic current. So we see that opposing neurotransmitters, GABA and glutamate, are packaged and functionally released together. The assertion that glutamate and GABA are co-released from single vesicle presumes that there is uniform vesicular transport and loading mechanism. But although they are released from the same vesicle, it does not mean that they are released in equal quantity, and it also does not mean that they are released at the same point of time. So there is temporal difference. Sometimes glutamate more, sometimes GABA more, sometimes both equally, but at different points of time. So what factors are important to consider in this differential release of glutamate and GABA from the same neuron? It is dependent upon the differential loading kinetics, vesicular pH difference. This is not dependent, this difference, and transporter activity via VGAT and VGLUT. So VGAT is a transporter for GABA and VGLUT for glutamate. So why do we need to understand this co-release of glutamate and GABA from the same neuron? So these points will try to shed more light on it. Understanding this co-release dynamics could reshape the pharmacological targeting of synaptic disorders. For example, for epilepsy, enhancing the synchronized inhibitory excitatory control may stabilize the circuits. In depression or schizophrenia, where GABA and glutamate balance is often disrupted, modulating the co-release might offer finer therapeutic tuning rather than just targeting GABA or just targeting the glutamate alone. Targeting specific transporters like VGLUT or VGAT, altering vesicular pH. So if the pH is changed, basically the release of glutamate and GABA differentiates. Modulating cytoplasmic calcium ions to influence co-release, for example, calcium channel blockers of N-type. Modulating transsynaptic scaffolding proteins to target vesicles structurally via gene therapy or CRISPR, and knockout genes for receptors. So basically, if we knock out the genes related to these transporters, we can modulate glutamate and GABA externally. We are now going to see some points regarding cancer because the genes which are responsible for glutamate aberrations are mostly the ones which are also found in multiple cancers in the body along with the metastasis too. So let us see the metabolic role of glutamate. Glutamate is a substrate for protein synthesis. So each point will try to relate with cancer. So if it is a substrate for protein synthesis, more glutamate means more protein synthesis and increased rate of growth of the tissue. Glutamate is a component of rigid B structure enriched in the polyglutamate. So the structural component of the cells increases, leading to enlarged tissue. Precursor for ornithine protein, so more amount of amino acids. A part of arginine cycle, and an inhibitor of glutaminase reaction. So if there is an inhibition of glutaminase reaction, there is increased production of decreased production of antioxidants, which leads to a further increment in the size of the cancer. Glutamine is also an energy source for some tissues. So basically, it will indirectly provide ATP to the cancer cells. And also a substrate for vitamin K dependent carboxylation of clotting factors. So leading to bleeding in the cancer that we see. We'll skip this one. Okay. So basically, this diagram shows healthy and cancerous patients and different organ systems. The only thing that we should know is the receptors present in healthy people and receptors present in cancer patients for glutamate are different. And this is a very stark thing that is seen in all the organ systems, further consolidating the role of glutamate systems aberrations in cancer. Now, role of glutamate in addiction. The aberrant glutamate transporter activity in VTA, nucleus accumbens, PFC, hippocampus contributes to addictive behaviors and drug seeking, especially by influencing the synaptic plasticity and neurocircuitry reinforcement. The below paragraphs basically just mention that transporters are not clearing glutamate well, and there is increased release of glutamate from the vesicles. So basically, the conclusion is we need to upregulate the transporters and inhibit the release of glutamate from presynaptic cleft. The review suggests upregulation and modulation of glutamate transporters as a promising anti-addiction strategy. It recommends future studies to investigate transporter-specific roles, especially VGLUT in relapse and withdrawals. This study was done very recently. In epilepsy, just like we saw a lot of glutamate in the cleft. So what will happen is there is an excitatory downregulation of signals. And if there is a lot of glutamate, lot of excitation, and if there is a synchrony of all those excitatory neurons, it leads to epilepsy. Hence, we need to look at epilepsy also from the glutamate perspective. At the molecular level, the hypothesis is plausible. GLT1 removes excitotoxic glutamate, and hence we need to regulate these transmitters also. Pharmacologic GLT1 upregulation via ceftriaxone or HSP90 inhibitors has been tried. We don't know the dose of ceftriaxone which is useful for upregulating these transporters. But it is proposed that low doses ceftriaxone for prolonged periods like 6 months or 12 months can be used for treatment of epilepsy. Selective receptor modulators, gene therapy for GLT1 in focal epileptogenic zones, and non-invasive brain stimulation to modulate astrocytic calcium dynamics, possibly altering glutamate receptor signaling. Going ahead with the intervention part, there will be focusing for 5 to 6 slides. Monoaminergic glutamatergic interplay. So many non-glutamate drugs also can be used to change the level of glutamate in the cleft. So ऐसा नहीं है कि we just have to use AMPA inhibitor or NMDA inhibitor, but there are other drugs also which do not directly act, but they have an influence on glutamate system. Drugs such as lithium, fluoxetine, they engage in the intracellular cascades, altering BDNF, mTOR, GSK, that restore the glutamatergic homeostasis. Transporter and receptor level modulation. So valproate and lithium upregulate the clearers of the glutamate, enhancing the glutamate clearance. Clinical relevance, so basically targeting glutamate indirectly offers safer long-term modulation, reducing the risk of excitotoxicity or hypofunction of glutamate compared to the direct actors. We'll see one by one regarding the various classes of drugs, what drugs can be used and at what places. So NMDA receptor antagonist. Overactivation of NMDA leads to calcium influx and cell death. But drugs like memantine, which is approved for Alzheimer's disease, basically blocks the excessive NMDA activity while sparing the physiological function. Same way for the ketamine. AMPA receptor modulators, selective positive allosteric modulators, may enhance the cognitive functions by modulating glutamatergic tone without excitotoxicity. So this strategy is currently under investigation. Glutamate scavenging systems. One novel approach involves enhancing peripheral glutamate clearance to reduce the CNS levels. Now, how can this be done? By enzyme-based therapies like glutamate oxaloacetate transaminase or glutamate pyruvate transaminase. So basically, we'll be using enzymes to clear glutamate. So first we saw that we can change the transporters or the receptors like AMPA and NMDA. Now we are going to use enzymes to change the levels, especially useful in stroke and traumatic brain injury. So we saw receptors, we saw using enzymes, and now the transporters, that is EAAT upregulation for clearance. So excitatory amino acid transporters clear glutamate from synaptic cleft. Drugs that enhance this expression of function, such as ceftriaxone, have shown promise in preclinical models of ALS and epilepsy. Glutamine and glutamate cycle modulation can be done. Ketogenic diet basically has also shown to reduce glutamate release and improve the mitochondrial function. This is because ketogenic diet already provides glutamine, so there is no need of excess production of glutamine, which can cause neuroinflammation. These are a few drugs along with their mechanism of action and their clinical use. So ketamine is there, memantine is there, acting on NMDA. Dextromethorphan and quinidine combination is there, which acts as an NMDA antagonist and also a CYP2D6 inhibitor. These are some unspellable names for AMPA receptor modulation. And these are basically for transporters. So riluzole, it acts on EAAT, ceftriaxone also acts on EAAT, and N-acetylcysteine, which increases the amount of antioxidants in the cleft, reducing the neuroinflammation. The drugs that we saw here, 1, 2, and 3, these basically acted directly on the glutamatergic system, but there are some drugs which act on the glutamatergic system indirectly also. So this table has those drugs, not just fluoxetine, but all the antidepressants, valproate, phenytoin, lithium, risperidone, olanzapine, aripiprazole, atomoxetine. Basically, by changing the scaffolding of proteins, by changing the synaptoplasticity near the cleft. That's it. So thank you for listening. Rajkumar sir.
Yeah, Dr. Aparna, you can take over for the question answer session.
Yes, sir. Thank you so much, sir.
Yeah.
First of all, thank you, Dr. Sagar. It was not only a very interesting and illuminating session, it was also a very lucid one. So not just, it's no surprise considering the ubiquitous nature of, glutamate in the central nervous system for it to be a part of multitude of neurodegenerative, neurodevelopmental, as well as psychiatric issues. But the connections with the various neurotransmitters, making them, the potential targets for intervention as such. And that is the most exciting part. And I think you've highlighted that very well. The floor is now open for questions.
Dr. Malebhai, if you can just chip in.
Yeah, so a couple of things. One is good presentation, so credit to you.
Thank
But
you,
try
sir.
and make your slides less populous.
Textual.
It is
Yeah,
too fine a print. Even on a large screen, it becomes a bit tedious to. So yes, information is necessary, but see to it that you're able to share information in a more easy format, not very. Now, apart from that, a couple of things. One is a very basic thing. I joined in a bit late, about 10 minutes into the presentation. And I heard the remaining part. What I wanted to ask you was, did you mention dimerization of receptors?
No, sir. Basically, I just showed in a diagram regarding various receptors that are present, not the dimerization concept.
Yeah, because dimerization is a more, of course, all concepts are very recent. The one you talked about, the same neuron having multiple neurotransmitter vesicles, and depending on the
Correlates.
pH and depending on the stimulation at a given point in time, which one will work. But what happens is that, see, we have understood, once the atypical or the second generation antipsychotics were available to us, that these are SDA, serotonin dopamine antagonists. So the simplest explanation that was given, and which somehow is understood or conceptualized to be the main working, is that when there is an increased inhibition of or antagonism of serotonin, we have we have dopaminergic activity which increases. So it's kind of a seesaw. But what people have understood over time is that there is something called as dimerization. So mostly, we have understood it with serotonin, dopamine. We have understood with glutamate and other monoamines that receptors, they also do not exist in isolation. Like we will have a D1 type of receptor, D1 subtype of dopaminergic receptor, which is actually structurally linked to one of the components of the glutamate receptor, NMDA receptor. So what happens is it's not the neurotransmitter activity that is important. Even at a very basic level, if let's say D1 is activated, it will kind of aid in the phosphorylation of a certain or activate a certain subcomponent of the NMDA channel. So you do not need any glutamate to be working on the glutamate, as you mentioned.
Mhm.
But even our traditional molecules have certain kind of activity like that. So this concept is important because this is more workable. And we have, you mentioned some of these. So this concept of dimerization is important, where there is structural connectivity of various receptor systems. Third thing, which I've always wondered is that, one, glutamate is widely present. It's not only present in the brain, but in almost every other organ system of the body. There is so much understanding of the glutamatergic system, but when it comes to translation, it is extremely tricky. In fact, very few agents, well, we can consider ketamine, we can consider memantine, dextromethorphan. That's about it. We do not have anything that is translated into meaningful clinical therapeutics. Yes, there have been other ones, riluzole and all that. Yes, they are used. But how much is their effectiveness in dealing with psychiatric problems? Amyotrophic lateral sclerosis is one. Uh, you know, uh, you mentioned epilepsy is one thing. See, all that is fine. We understand that basic mechanisms of illnesses, neurodegenerative disorders, neurodevelopmental disorders is same. But the question that is, which is going to be troublesome is, why do some people have only symptoms of psychosis and not epilepsies? Why do some people have epilepsies but they do not have OCD? Why are some people who have everything? Because when you have a system which is widespread, and if you see at a real microscopic level, there don't seem to be much difference between what happens in OCD, psychosis, mood disorders, everything is same. But if we say that bipolar is same as psychosis, then why are there mood fluctuations? In the sense, why are there episodes of mania and why are there episodes of depression? So in spite of knowing a lot about glutamate, it keeps on advancing, we still are unsure when it comes to therapeutics. Fourth point is that the glutamatergic system, especially the NMDA receptors. NMDA is a complex receptor. There are 7 subunits, and it requires it's a magnesium block, and there are multiple agonist sites, and it's a chance it's a chance receptor, so to say, because unless 3-4 things are in place, it doesn't open. Unlike AMPA, which is fast, right? And once it opens, it doesn't close. So there are subcomponents of the of this NMDA glutamate receptors. These are the NR1, NR2, NR3, those kind of subunits which are there. Of which, even as we become older, right from intrauterine development till adulthood, there are changes. So some subcomponent is present only in utero. And that subcomponent is not there when we see or when we investigate disorders, whether it's in human beings or in preclinical models. So pretty complex system. And whatever we know, it's kind of, each time you read about it, there is a layer of complexity added to how things work, how it interacts with other systems. And probably that is what makes it interesting, but at the same time, very difficult. There have been a lot of studies, a lot of those alphanumeric agents that you mentioned, which have been also tried for the metabotropic glutamatergic receptors, which have not really yielded anything. Even phase 1, phase 2, and they're out. It doesn't even go to phase 3. So that this is where the thing is. So good, lot of food for thought. Sir, if there are any questions, we can take them.
Yeah. Dr. Aparna, can you take the questions?
Yes, sir. We have a question from Dr. Vishnu Priya. Can we use any of these NMDA mediators in ASD?
Sagar.
Hello?
Hello?
Yeah, Dr. Sagar? Yeah.
Yeah.
Yes. So yes, theoretically, as Melissa told, theoretically, basically there are drugs which we can use to alter the glutamatergic system, especially in ASD, for which we need to understand what is wrong in ASD. So three, four things. First is the glutamate-glutamine imbalance is present. Secondly, AMPA receptors are in a deficit. And because of this, there is the neuroinflammation present in the synaptic cleft. So theoretically, because of this neuroinflammation, there are social deficits and stereotypic behavior, and for which memantine can be used. But then there are no trials currently which have proved that memantine can lead to reduction in stereotypic behavior or reduction in the social deficits.
Thank you, Dr. Sam. Dr. L.C. Sunda has a question that is there any role of glutamate level in production of negative and positive symptoms of schizophrenia? And also, how does it affect dopamine levels at the synaptic?
Okay, regarding schizophrenia's positive and negative symptoms, positive symptoms are easy to explain because there is more glutamate in the cleft, more stimulation of postsynaptic neurons, leading to excitation, and excitation is basically translated into delusions or hallucinations. But it is the negative symptoms that we need to understand in a different way. Excess glutamate basically needs to be cleared off, and that requires a lot of antioxidant action. And when that does not happen, that inflammation continues for a prolonged period of time. And prolonged inflammation then leads to these negative symptoms. So these drugs in a long-term use theoretically should help in the negative symptoms of schizophrenia.
And how does it affect the dopamine levels at the synaptic system?
I'm sorry, I think Malay sir might be the right person for this question.
No, I think you did mention, no, that see what happens is in a very simplistic model, there would be both effects on the dopaminergic system. There would be a stimulatory effect as well as an inhibitory effect, which is in balance. The stimulatory would be direct. You have a dopaminergic neuron which goes to the midbrain and substantia nigra and stimulates dopamine release. That is the basic effect of glutamatergic excitation. But at the same time, glutamate also stimulates GABAergic neurons because we have this interneuron kind of thing, no? So one of the type of neurons which you mentioned was a parvalbumin interneuron. This parvalbumin rich interneuron is one of the key anatomical substrates in schizophrenia pathophysiology. Because if it doesn't work, then the key regions that we understand are implicated in the pathophysiology of schizophrenia do not get inhibited. They keep on getting stimulated all the time. So what, in simple words, you have a direct effect. So from cortex to the midbrain, excitation, glutamatergic, dopamine activity is more. So that is one arm. And the other arm is that these interneurons will also stimulate GABAergic neurons. So once GABAergic neurons are stimulated, it will provide inhibition. So that loop is maintained. It's a negative feedback loop. So when we have in schizophrenia, let's say too much of excitation, okay? If you have too much of excitation, what happens at the base, at the midbrain level is dopaminergic overactivity. Now, if there is a problem with the interneurons, okay, and they do not get stimulated enough, or they are overworking, then we will have undue inhibition of the dopaminergic currents to the cortex, which would be negative symptoms because anhedonia, amotivation are cortically defined in terms of what the locus of action is, rather than mood symptoms and things like that, which are more of limbic, right? So basically, we need to know how much is, if we are looking at dopamine, what is the sum total of dopaminergic activity in the prefrontal cortex? D2 is all low level in the sense the traditional midbrain substrates, locations which will produce the positive symptoms, delusions, hallucinations. because as you also mentioned that वो जो ऊपर signal आता है, the signal to noise ratio is pretty low. So you have got more noise and lot of unnecessary analysis of input coming from the low regions and misidentification, misinterpretation, misanalysis, mislabeling, and all those kinds of things happening. But in the cortex, if you have a sum total lesser dopaminergic activity, which is mediated by D1, D3, all these kinds of other ones, if that is less, and that is also glutamate, I mean, not only, but one of the main components of that is glutamatergic. This will lead to a progressive hypodopaminergic state. So lack of motivation, lack of involvement, that is apathy, evolution, and all those kinds of A's that are there in the negative symptoms. This is how it works. But the basis is not as simple as this. That is why I asked you whether you mentioned this dimerization. Because if you have two receptors which are like tied, two different systems which are like this, then something which is act on one is also going to have an immediate effect on the other. So this is what is this is how we understand. As I said, if you are doing the same topic next year, we'll have something more.
Thank you, sir. Dr. Vaibhavi has a question that modafinil also has glutamatergic action. Is there any scope to expand its use apart from the current indications?
So basically, modafinil can increase the glutamate levels in hippocampus, in thalamus, and PFC also. So depending upon the area where there's more glutamine, we can use modafinil. Like if there is hypoactivity in PFC because of low glutamine, that is where we can use modafinil. So basically in ADHD, so modafinil might theoretically help in such cases. Or cases where there is prolonged negative symptoms in schizophrenia or depression. That is where minor increment in glutamate through modafinil via AMPA and NMDA can help. This is my understanding of using modafinil in ADHD, negative symptoms of schizophrenia, depression.
Yeah, correct. So where we require some kind of more of stimulatory activity, okay. Again, the thing will be this, that when you look at ADHD or when we look at negative symptoms, whether the same circuits are involved in all these things. And even if the same circuits are involved, whether they're involved in the same intensity or frequency, that is important. So what also happens is that when we give for ADHD, when we give something like modafinil, they might really activate. It might not only cause an increase in attention or a cognitive improvement that way, but it can also lead to more of glutamatergic activity. And if there is a predisposition to developing psychotic symptoms, a person can develop psychosis. And this is applicable across. It's not only glutamatergic. Because see, the problem is this, that the molecules that we have are supposed to be I mean, if we look at the fact that most molecules in psychiatry are designed by serendipity, means we have nothing to do. We just found out that somewhere it works and the other thing doesn't work. So we found out and we are happy with that. The problem is that after that, what has been devised, the molecules, let's say, of the early 21st century, so last 20 years, are more designed. People have thought, okay, let's target the glutamatergic system, or let's target this or that and see what happens. Designer kind of drugs or something. The focus is again on one system only. And believe me, none of the symptoms in none of the illnesses in neurology and psychiatry are one system based. Okay, we need to look at it. So we have one molecule. And again, the design of glutamate in the brain is such that it works at different levels in different ways. But a molecule will work in the same way. So even if you look at typical antipsychotics, the prototypical example of haloperidol D2 blocker, fantastic results with delusions and hallucinations. But it randomly blocks, not randomly, it blocks all kinds of dopamine receptors. So it blocks D1 in the cortex. So very, secondary negative symptoms. If you use for mania, depressive symptoms. So it blocks everything. But glutamate and GABA don't work that way. They have got very fine-tuned mechanisms in the cortex, in the basal ganglia, in the midbrain, in the spinal cord, and so on. Now, for us, drug design does not reach that level. So we keep on struggling. So we need to understand that molecules will have widespread effects. Not only on neurotransmitters, but they will have widespread effects on systems also. So whether that system is inflammation, whether that system is endocrine, whether that system is ABCD, XYZ, there are going to be some effects. The question is whether these effects are adding to the proposed therapeutic effect of a molecule or not. So if you see that glutamate, you will have what molecules do we have that are really glutamatergic in nature at the moment? So these ones which are there specifically. But the ones that we use, many of them have anti-glutamatergic or pro-glutamatergic properties. The question is that is what we have to figure out, is whether this glutamatergic activity of existing molecules that we use day in and day out is significant or not. And there I think you have, I think you mentioned that one study where people have found that when people used whatever
Yeah, and
agents,
post antidepressant,
yeah, so there was hardly any change.
no change. Yeah.
Yeah. So one study, but yes, this is where we are going to the I mean, we have got information to work on. We can have our own hypothesis, but that doesn't mean that we are anywhere near the truth. We can try permutations and combinations. People have tried, people have thought that this will work, that will work. Yes, in some people it will work. Whether that would be a recommendation for everyone, maybe not.
Thank you so much. Are there any other questions?
No, I think not.
Yeah, I would
Okay.
hand over the floor to you, sir.
Yeah. Thank you, Dr. Sagar. Thank you, Dr. Arpana.
Thank you, sir.
Thank
Thank
you, Malai. And thank you, friends. The next 6 sessions are going to be much more stimulating, with Malai taking 6 sessions consecutively. So we would be looking forward to again in the second Wednesday of June. Till then, goodbye, good night.
Thank you.
Thank you. Thank you, sir.